This lecture is coming to you from the testing center, which is where I was at the last period of the day. It's eerily quiet in here without students, so I'm going to fill it with noise, and let's get through this lecture. So today we're going to talk about the structure of genetic material. We'll talk about DNA replication. The stuff that's coming down here will be a part of genetics, but this will be a little bit later on. So our main focus is the structure of genetic material and then DNA replication. So that'll lead us to a pretty good essential question or set of them. Pretty easy. We'll have two of them, but the first one's what is the structure of DNA? And then we'll have a second one. Here, we'll put one. The second one is how does it replicate? And if you don't like, with the whole English thing, if you don't necessarily like using it because it's not specific enough, how does DNA replicate? All right, so let's start with this structure, and this gives us a little history. So it's not quite as important to get the history, but at least we'll get some background knowledge. So early in the 20th century, the molecular basis for inheritance was a mystery. Nobody really knew how it happened. Biologists did know that genes were located on chromosomes. We did know that. But it was really unknown whether the genetic material was proteins or DNA. So biologists finally established the role of DNA in heredity through experiments with bacteria and viruses. So that's why this kind of gets to be a cool section where we talk about viruses. It really was a breakthrough in the field of molecular biology, the study of heredity at the molecular level. Which makes sense, biology, heredity at the molecular level, it's molecular biology. So here's some important characters that might be worth knowing. In 1928, we had Frederick Griffith, was surprised to find that he killed a pathogenic bacteria, which mixed with bacterial remains, with living harmless bacteria. Some of the living bacterial cells became pathogenic. So what he's saying is that we have a bacteria that we've killed. And then we have a bacteria that's kind of like a gut bacteria. It's a good bacteria. And so what happens is when he mixed the dead bacteria with the good bacteria, some of the good bacteria then became like bad bacteria. I don't know, I'm trying to make, I don't know, what's an angry face or whatever. It's not working, but whatever. Angry face, wait, I messed that one up. There we go. Okay, forget that one. There we go. So, dead bacteria plus good bacteria equals angry bacteria. So, all of the descendants of the transformed bacteria had inherited the newly acquired ability to cause disease from this guy. So, in 1952, we had Alfred Hershey and Martha Chase used bacteriophages to show that DNA is a genetic material of a T2, a virus that infects the bacterium of E. coli. So bacteriophages, which we end up calling phages for short, are viruses that infect bacterial cells. We'll look at them. They're kind of alien-looking creatures. Okay, phages are labeled with radioactive sulfur to detect proteins and radioactive phosphorus to detect DNA. So bacteria was infected with either type, a labeled phage to determine which substance was injected into the cell. So the whole point of the Hershey-Chase experiment is really to figure out what's causing, what's the genetic material. So they had sulfur-labeled protein, so with a little sulfur label, and then on the outside they had DNA, which was labeled with a phosphorus, to detect inside the cells. So cells with phosphorus-labeled DNA produce new bacteriophages with radioactivity in the DNA, but not in protein. I know all that's really confusing, so here's a little bit better of a thing. So here's a bacteriophage, by the way. Bacteriophage. Okay, so here he is. He's got two parts. The yellow or kind of gold color on the outside is protein. Viruses are very, very simple creatures. They're protein on the outside, and then the blue part on the inside is DNA or genetic material. So what this experiment said, let me see, do we have a picture? Oh, we do, yay! So what this experiment says is that we label, basically we're labeling the DNA, or the protein. So we put a little sulfur thing on the protein, so all the gold parts. And then we swish it around, we let it infect cells, and what they found out was all of these cells did not have any of the proteins in them. So the cells, which would have been in what we call the pellet, did not contain the radioactive sulfur. So no go there. So then they used a different label. This was radioactive phosphorus. They put a little phosphorus label on it. And so they tracked that. They swished it all together, let it infect those cells, and they noticed in this pellet, so in the actual cells, that's where the DNA was. So they could say, okay, based on these like simple models, we can tell that the genetic material is based on DNA, and DNA is the part that's carried on and what kind of causes these cells to be infected. So basically that's the whole point, is that this experiment shows that DNA is the genetic material. Okay. May sound obvious to us, but don't take that for granted, because scientists back in the day had no idea. And imagine, how would you figure it out that DNA was the molecule that was our genetic material? How would you figure that out if you had no idea? So these are what these guys did. All right. We'll talk more about viruses later and how they affect things, so I'm not going to worry about that. This hopefully is at least a little bit of a review. We have DNA and RNA, and these are our nucleic acids, where we get the Na from in both of them. They're nucleic acids. And they consist of long chains or polymers of chemical units, our monomers. Remember, our many mers are made up of one mers, and they're called nucleotides. Okay, so our monomers for DNA and RNA are nucleotides. One of the two strands of DNA is called a DNA polynucleotide. It is made of many nucleotides. Say it's not, they're not creative here. So it's just a nucleotide polymer. A many nucleotide sequence is one strand. See, one of the two strands. So nucleotide, as we talked about before, is kind of composed of a nitrogenous base. I'll draw them for you a little bit. So let's say that this part's in A. Then we have kind of a 5-carbon sugar, a little pentagon here. So this would be, let's make DNA. So this is deoxyribose. So that's our 5-carbon sugar. And then stuck to that as a phosphate group. Boop. So that's essentially what our nucleotide looks like. The nucleotides are joined by sugar phosphate backbones. There'd be another phosphate here, another sugar stuck down there, and that's our little backbone. So as a better picture here, sort of better. Let's look here. Okay, so these would be the little phosphate groups. Gotta draw bigger than these spaces are. These little 5-carbon sugars, notice they've got little, like, they're little pentagons, so that's how we know our 5-carbon sugars. And those guys attached repeatedly, kind of phosphate sugar, phosphate sugar, phosphate sugar. So they make up the whole kind of backbone that we see, the whole kind of like railing of the staircase of the DNA molecule. And then out from those, we have the things that we know of as our nitrogenous bases, those kind of identifying sequences, our A or C or T or G. Okay. And so they make up kind of those rungs of the ladder, if you will. All right, so a lot of stuff going on here, but we've talked about it a little bit. So if you still have trouble and don't understand, I've got models in the classroom, so come by during flex or tutoring and we'll take a look at it again. Okay, we can do this very quickly. Each type of DNA nucleotide has nitrogen-containing bases, A, C, T, and G. All right. Hopefully we remember those guys, adenine, cytosine, thymine, and guanine. If not, we shall review. They have some different characteristics. So there's the pyrimidines, and they're a little bit shorter. And we have purines, and they're the larger molecules. Somebody thought of this one as like, if you think back to chemistry, how you can tell them apart is if you have pure silver. So it's purines, or A and G. Isn't that magical? That is actually helpful for me. Pure silver. All right. So that would probably be a good place there. Hopefully a little review. But if you need to, this is a good place to stop the video, pause, write some questions out on the left-hand side, and then get back to it. All right, to review, the full name of DNA is deoxyribonucleic acid. So the nucleic is referring to the location in the nuclei of eukaryotic cells. Sorry for the squiggly line there, but in the nuclei. RNA, ribonucleic acid, okay, is unlike DNA. So this is a good one. This is one of those things that we're going to need to know kind of as we continue. So it's unlike DNA in that the sugar is ribose instead of deoxyribose. So we have a different sugar. Okay, and also we use the nitrogenous base of uracil. And there's a like sciencey, nerdy, kind of cheesy pickup line that says like, if I could choose, I'd be RNA because it has U in it, or I'd have RNA because it has U in it. Something silly like that. Find the meme. Hey, there you go. Anywhere in this lecture, let's do this. We'll do a totally new. This is a experimental. For your quiz to get points, send me an email of a meme that has to do with anything from this lecture. So it could be RNA, it could be later when we talk about DNA replication, doesn't matter. Send me a meme. That's how you get your 5 points. Send it to me in email. Send it to my Gmail account. All right. Maybe I'll use them. I always need to update my memes, right? Okay, so here's our difference. Again, phosphate group. Again, nitrogenous bases, but with a slight difference of uracil, the U, it has U in it. And then the sugar is still a 5-carbon sugar, but it's a ribose instead. Okay, a little more history here. So after the 52 Hershey-Chase experiments convinced most biology that DNA was the genetic material, a race was on to determine what the structure of the molecule was so it can account for its role in heredity. So researchers focused on discovering the 3-dimensional shape. And you don't have to write too much here, but we'll look at it. So American Watson, James D. Watson, journeyed to Cambridge University in England to work with Francis Crick, studying protein structure in a technique called X-ray crystallography. While visiting a laboratory, Maurice Wilkins at King's College in London, Watson saw an X-ray image from Wilkins' colleague Rosalind Franklin. So Rosalind Franklin actually did the X-ray crystallography. That's sort of what it looks like. She didn't get kind of up for the Nobel Prize until after she died, and you don't get a Nobel Prize, and you know, if you're already dead, the posthumous is what they say. So she didn't get one, but she is, just so you guys know, she is a boss, and she is like the underlying determinant for what the structure of DNA was. But back in the day, females did not get a very big role in science at all. So we have come a long way as scientists. So Watson, oh, by the way, I got on my little tirade there and forgot to show you the picture. So the important part here is this box. So you see kind of the circular thing. What she imaged was the little like rungs. So you kind of have to, we could look at it in the classroom, but it's like if you took DNA and you always see the ladder structure kind of from the side. This is going to be atrocious. Okay, whatever. It looks like this. If you took the look down this way of that 3-dimensional shape, that's what you would see. So Watson and Crick took this image of a spiral of this helix with uniform diameter, bases located above one another like a stack of dinner plates. The thickness of the helix suggested that it was made of 2 polynucleotide strands. So Watson and Crick deduced this shape based on this image that they saw. So they realized DNA consisted of 2 polynucleotide strands wrapped in a double helix. The sugar phosphate backbone is on the outside, which we've determined, so we know that. The nitrogenous bases are perpendicular to the backbone and in the interior, like rungs. The specific pairs in a helix form are A's with G's, so straight letters with straight letter, curvy letter with curvy letter, so G's with C. Okay. And they form a different amount of hydrogen bonds, but I don't really get into that too much. Okay, so here was them working through the whole thing. And just so you know, I will put it as diplomatically as possible. Watson is not a very likable guy. Okay. And there you go. But that's their basic idea, is that we have a ladder shape, and then we start twisting it. And that gives us the helix shape. Okay. The whole idea that we're looking for is they're bonded up and matched. So although the whole idea with purines and pyrimidines is that one is larger than the other, but they always match in a form that the large one and the small one match. So this distance here is always the same. Okay, so the distance is the same here as it is down here. Okay. So that's the idea. And still, we take that and you twist it up and you get kind of the model. So you could get ribbon model or the computer kind of space filling model. Shows us the same thing. So in '62, the Nobel Prize was awarded to James D. Watson and Francis Crick and Maurice Wilkins. Rosalind Franklin probably would have received the prize as well, but her death from cancer in 1958. Nobel Prizes are never awarded posthumously. Watson and Crick model gave new meaning to the words genes and chromosomes. The genetic information in a chromosome is encoded in the nucleotide sequence of the DNA. So we got good stuff from them, even though Watson's a jerk. I didn't say that, but it's true. So we're at 17 minutes, but maybe it is a good place to stop, take a break, review, chunk your notes a little bit if you need to, ask some questions, and then come on back. So part 2 of our discussion involves DNA replication. So a whole big thing is that DNA has to make copies. When we talk about, here's a little introduction or a little review here, if we go back and make it for you. Remember, we have G1, we have the S phase, we have G2, and obviously this isn't to scale. God, it's terrible. Anyways, we have like mitosis and cytokinesis. Just bear with me on it. But remember, in order to undergo mitosis in the cell cycle, we have to copy the DNA. So in this S phase, we actually have to undergo DNA replication. So this actually applies to the cell cycle here. So think back to that when we go forward here. So the DNA replication follows what we call a semi-conservative model. Semi means half. Conserve means really to keep the same. When we talk about conservation efforts, we're trying to keep an area the same. So we're keeping it the same. So half is kept the same. So the 2 strands are going to separate, and each strand then becomes a template for the assembly of a complementary strand from a supply of free nucleotides. So each new DNA strand has one old one and one new strand. So here's our old strand. We've unwound it because we could. So what we'll end up doing is we're going to kind of break it apart. And so each of these halves, then let's make some color here, becomes a template for another half. So this is why we call it semi-conservative, because half of it is in fact kept the same. It's the same as the original kind of over here. All right, let's go back to blue. I like the blue one better. Okay, so if you didn't get my squiggles there, the parental DNA molecule looks like this. Okay, we'll separate those guys down the base pair there, and then we get these free nucleotides, which will come in and find kind of and match up and line up, and then in the end we'll get 2 identical molecules. Now we always note them for color, but there's not going to be any color difference in the actual cell. If you look at it, we've got A and T, C and G, C and G, GC, GC. So the whole point is that these guys are exactly the same. Okay, exact same. Oh boy, hopefully you can understand that squiggle. All right, it looks a little bit more complicated in the actual DNA molecule because remember that it's wound up. But you can sort of see what's happening is that we unwind at a certain portion, and then each of those kind of ribbons of the DNA matches up and lines up a new segment. So we'll end up getting two exact copies. It may not look like it, but we've got A and T, followed by CG, CG, AT, AT, and AT. So we're getting exact copies of each. That's why it's a replication. We're making replicates. So replication of a DNA molecule begins at a particular sites called origins of replication. That's where it starts. There's short stretches of DNA having a specific sequence of nucleotides. Proteins that initiate DNA replication will attach to the DNA at the origin of replication. So we have some proteins that will start it. Okay, they'll attach at the origin of replication. They separate those two strands of the double helix. Okay, and they'll produce. Then they'll proceed in both directions, creating replication bubbles. So sort of what that looks like is here are different origins of replication, and so we'll get little proteins to attach at those spots to pull them apart, and then replication will happen at both sides. Notice they look like little bubbles until eventually those bubbles will kind of meet up, we line up, and notice we'll get two strands, and they will be exactly the same. Okay, so DNA replication occurs in a 5' to 3' direction. This is going to be one that we'll kind of have to work through together because it's a weird one to see. But DNA replication occurs in the 5' to 3' direction. We could get into it, but honestly, it's an organic chemistry thing as to why they're named that way. But it goes 5' to 3'. And I'll show you what it looks like here in a minute, or at least point it out. So replication is continuous on the 3' to 5' template because they're actually opposite. So one strand actually goes this way, and this would be the 5' end, and this would be the 3'. Well, then this one, runs the opposite direction from 5' to 3'. So it's really weird, but basically, on one side, we can run continuously. We'll just add one after another. Okay, the DNA polymerases add nucleotides only to the 3' end of the strand, never to a 5' end. They're not made that way. By the way, this is a DNA polymerase. It's an enzyme ACE that makes a polymer of DNA. So replication is discontinuous, so it doesn't go one after another on the 5' to 3' end. I know, it's lots of things. We're going to kind of work through it together. This part especially is where it's like, what are you talking about, these numbers? We're going to look at it and try it together. So an enzyme called DNA ligase will link fragments together. Those fragments were found by, I think, a couple called Okazaki. So they have entire fragments named after them in the DNA. It's the coolest thing. All right. But it will have a DNA ligase that links or ligases those pieces together. So basically, it's numbered this way, and it doesn't make a whole lot of sense. Just don't ask a whole lot of questions on that one. You just got to take my word. If you're really like, I want to know the nuances of that, take organic chemistry in the future. It will make you happy. It did not make me happy, but that's okay. So here's our 5' end, and this is our 3' end. So if we're running a DNA polymerase right here, and it's attaching the pieces on, it can do it from 5' to 3'. So it can go this way easy. Okay, which means our 3' to 5' end is a good template kind of. But it's going to work weird. But this is, anyways, they're going in opposite directions. That's the whole idea. This one runs this way. This one runs this way. I'm getting ahead of myself. Okay, they go opposite directions. They're pointing separate directions. So 5', 3', 3', 5'. So more or less what has to happen, and we'll talk through these, is that you've got a 5' in here, 3' in here. So the DNA polymerase can actually run 5' to 3' nice and continuous on this strand towards what we call a replication fork, where we're splitting those. It can't run this way. This is not a possibility from 5' to 3'. So what this guy actually has to do is it'll jump to a segment like right here, and then it will, let me change colors. It'll start there, and then it'll actually go this way. So then it'll jump back with some molecules and then run this way. And then jump back here and run this way. Each time, these things have to be kind of filled in together. But one segment can go continuously, just one right after another. The other one has to jump back each time and go continuously. The idea is that overall, everything's running this way, but this thing's going to have to jump back this way each time that it wants to do something. So this is what we actually call, oh, we don't have it, my bad. Okay, so this is up here, this one that's working so nice and smoothly is what's called the leading strand. Okay, and this one at the bottom is what we call the lagging strand, because we got to jump back, and so it lags a little bit. So DNA polymerase and DNA ligase will also repair itself. Oh, here's a fun meme for you. I love this one. It's DNA checks itself before it wrecks itself. So DNA polymerase will also repair DNA. They're checking and making sure that that DNA is not wrecking itself. Okay. DNA replication ensures that all somatic cells, somatic, if we haven't talked about, are non-sex cells. Somatic or everything that's not egg and sperm, basically. Okay. So it ensures that all somatic cells in a multicellular organism carry the same genetic information. We're making exact copies. Okay. So that's actually the very, very quick, rough version of that. So if you have questions or don't understand exactly what I'm talking about, which is fine, you may not, we're going to cover it in class. Do your best to try to look at it. There'll be some videos. We'll work through it. It's coming. But if you have any specific questions before class, feel free to shoot me an email or come by tutoring or flex time. I'm around. All right.